Hi guys, this is Divya Pradhan again, and we are going to talk about the scientific method. Well, when we're talking about science, science is an organized body of knowledge consisting of demonstrable truths obtained by careful observation, experimentation, and reasons. Remember, it has to be demonstratable. That means you just can't say something and not be able to prove it. Whatever data you have collected, somebody can collect the same data and come to the same result. Science, of course, therefore, is a process, and the process of science is called scientific method. Not a big deal. All right. Aristotle is considered to be the father of scientific method, and in making the scientific method, there are certain number of steps. First is making an observation. Second, formulating a hypothesis. Third, performing experiments, observations, collecting data, and then finally forming a conclusion. And once you make the conclusion, then you make a theory. All right, so the first step is an observation of insight that makes you curious. Say, for example, you are curious or you want to understand more the process of diabetes or cancer or pneumonia or typhoid, anything that interests you. What you do is, all scientists, once they choose a topic of interest or something catches their attention, they start gathering information. And when how they gather information, of course, is from libraries, conversations, internet, etc. And of course, what happens is, once they do that, then what happens is they form a hypothesis. A hypothesis is nothing but an educated guess. Say, for example, hair texture attracts me. So I go and look up all the information on hair. Well, how do I make my hair more silkier? Or how do I make my hair more thicker? Or how do I improve the quality of my hair? So then I look up information and I say, oh, I find all this cool natural stuff online that helps hair texture or hair color or hair shine or hair quality. Well, and then I say, okay, putting egg white on hair helps to make the hair more shiny or makes my hair more thicker. So that again is a hypothesis because that has not been proven yet. All right. So here this is something called inductive reasoning, because you have collected information only. You have not done any experiment. So this is called inductive reasoning. You have induced from information that is already available. Okay. Well, of course, the third step is you do an experiment. But whenever you're doing an experiment, remember there are three things you have to have. You have to have a control group. You have to have an independent variable. You have to have a dependent variable. First of all, what's the independent variable? Independent variable is something that you are changing. Let's say, for example, I found this miracle drug, let's name the drug X, that I know can help with diabetes. So what my hypothesis is that as long as X is given to diabetic patients, well, their condition will improve. All right? So here a set number of diabetic patients are given the drug X. So that is my independent variable. That is my variable that I am changing. I am manipulating that. Dependent variable in this case will be, of course, again, because we are checking for diabetes, will depend on the blood sugar level. Because with diabetes, as you know, the blood sugar level becomes very, very high. So dependent variable is what is changing because of this independent variable that you have changed. So you are going to have two sets of group. One, you're going to give this drug X, and one group you're not going to give the drug X. So that's my control group. So control group is this, I'm giving everything else to these diabetic patients, but I'm not giving the drug X. All right, that's my control group. This is the group against which I'm going to compare my results. Now, of course, what happens is I will collect data from my experiment. And once I collect my data, based on the data, I will prove either hypothesis is either accepted or rejected. If my drug X indeed reduces the blood sugar level, I'll say, wow, this drug X works for diabetic patients. All right. Say, for example, the drug X did not change the blood sugar level in comparison to my control group. Then what I say is drug X is not good, and therefore what happens is my hypothesis is rejected. And this is how scientific theories are made. Okay. So here's the flow chart. Like I said, observation. Remember, observation is that when you read and then you form a hypothesis. This is, remember what this is. This is inductive reasoning, because you've induced, then you can conduct experiments. You can, once for conclusion. Now this will be your deductive reasoning. By the time you reach here, you're deducing from your own experiments. You're saying, okay, this drug works. So this is deductive reasoning. The G is missing in there, so I'm going to put it in here. And therefore what happens is you come across a scientific theory. So what hopefully your understanding is, scientific theory, yes, today it holds true, but tomorrow it may not hold true. Like, let me give you an example. When antibiotics came, well, a lot of diseases, bacterial infections, could be controlled. But look now, a lot of bacteria have become resistant to antibiotics. So now what happens is that scientific theory is changing. So now, for example, MRSA is Staphylococcus aureus, which is a bacteria, has become resistant to the drug methicillin. So now what do? Well, so now we have to use other antibiotics like vancomycin. Now that what I'm trying to explain to you is scientific theory or scientific methods or scientific theory are always changing. So here's an experiment. So here you have, this is my control, of course, that there is no water. And this one has, here what happens is there's no water. Here it has all this is all. So it has water, it has light, it has air. Okay, so here is, of course, my control. I have everything in here. Here, this is my, what am I changing? I'm changing water. Here, what am I changing? I'm changing light. Here, what am I changing? I'm changing air. So here, notice that my independent variable is what? Water. Independent variable is what? Light. Independent variable is what? Air. What's my dependent variable? Dependent variable is the growth or the germination of the seed. Okay, so here we see no germination. Here we are seeing no lights. Okay, so germination, good amount of germination. So here we are seeing no air. So here you have poor germination. So here in the control we have water, light, and air. So what can we conclude? We can conclude that germination is best. What happens is when there's no light, when we have water and air. This is how experiments are conducted and theories are made. Alright, so some of these standing biological theories that we have is cell. All organisms are composed of cells. Okay, and new cells come from pre-existing cells. They don't come from air, they come from pre-existing cells. Gene organisms contain coded information in the form of DNA or RNA that dictates their form, function, and behavior. Evolution. All living things have a common ancestor, but each is adapted to a particular way of life, and we are evolving. Homeostasis. The internal environment of an organism stays constant within a range protective of life. Ecosystem. Organisms are members of population that interact with each other and with the physical environment within a particular locale. So these are standing theories so far. Remember, in the next, I cannot guarantee you that this will not change. In next 25 years, who knows? We can form new cells from no pre-existing cells. So that will totally topple our biological theory of cells. All right. So, like I said, as for now, these theories have been developed based on those steps. If there are any questions or concerns, please contact me. And with this, we come to the end, and I will see you next time. Thank you.